RNA-Seq is a technique used to sequence the transcriptome of a given cell or tissue type. The transcriptome is the complete set of transcripts and their quantity that are made in the cell. This gives information to that particular coded RNA, which includes the quantity, and tells expression of a given development or physiological condition. RNA-Seq is made possible by next-generation sequencing. Why look at the transcriptome? All cells in the tissue have the same DNA sequence, but how the DNA is expressed, meaning which parts of the DNA and how much is transcribed, is how one cell type differs from another. Knowing the transcriptome can help scientists determine if a gene is active in the particular cell or tissue type. This can give an indication of the gene's function, meaning if a gene is not expressed in healthy tissue, but is expressed in large quantities in a diseased cell, then that gene has likely something to do with that disease. This is how knowing the transcriptome can help understand how gene expression contributes to disease.
Since RNA-Seq shows the unique gene expression of your gene, by doing this you're able to discover your gene of interest, unique expression, which in turn equals your gene's unique transcriptome. With measuring the unique transcriptome by doing RNA-Seq, you're able to discover more RNA characteristics about your messenger RNA. To perform RNA-Seq, there are 6 basic steps to follow. First, you must isolate your RNA. RNA isolation for RNA-Seq is no different than doing RNA isolation protocol that we perform in class. First, you must homogenate or mix your tissues. You can do this by using beadbug homogenators shown on the left or the Downs homogenators shown on the right. Next, you must precipitate out the top layer in the tube without getting the middle layer, which houses the DNA. This top layer is what houses your messenger RNA. Step 2 is to turn your messenger RNA into cDNA through reverse transcriptase. Since RNA is easily degraded by RNases, which are found everywhere, it is easier to work with cDNA. Once you produce cDNA, it is cut into tiny fragments for easy sequencing in future steps.
The cDNA fragments must undergo additional processing in order for them to go through next-gen sequencing. The first step of that was the fragmentation of the cDNA into small pieces of sequence. The next step is to add adapter oligos for the process of the next-gen sequencing machine. The oligos are represented by the yellow and white segments of the cDNA fragments. These are added on the ends of these fragments in order for them to be able to be sequenced by the machine. The fragments can then be rapidly sequenced due to the advanced fluidistics of the machine and the short length of the fragments. Base pairs added to the template strands of the original sample are read by the processing of the tagged nucleotides that have been added. So fluorescence is what actually reads these sequences of the small fragments. The computer then processes the sequences of the fragments and aligns them with each other using base pair complementarity. Once all of the fragments have been aligned, the transcripts have all been sequenced. This collection of sequence is called the RNA library.
Once an RNA library has been established, the library can be compared back to the genomic DNA sequence in the form of those overlapping fragments, which can be combined into contiguous sequences corresponding to the coding sequence of the transcript. Using RNA-Seq, the exon boundaries can be determined to the nucleotide. The last step of RNA-Seq is to quantify the transcripts of any given cell, tissue, or organism. This is done by determining the frequency of the nucleotides within the transcriptome. The frequency of each nucleotide is plotted. You can see that exons of the isoforms give the corresponding nucleotides a frequency. Here, we see a decrease in the expression level of the nucleotides because isoform B does not contain them. All the expression seen here is due to isoform A. RNA-Seq can be used in research to map gene and exon boundaries as we just described. It can be used to identify novel transcripts with such low expression other methods cannot detect them. It can be used to monitor gene expression throughout an organism's lifespan, as the Modern Code project has. Modern code has specifically been responsible for determining the expression level of all genes in Drosophila and creating the RNA-Seq track you will use to annotate your contig this semester.